Good morning, ladies and gentlemen. It's my great pleasure to have my talk in 2014 APCIS. Today, I'm going to review main steps of cardiac development. The adult heart have two atriums and two ventricles with separate circulation. Initially, the primitive linear heart tube forms by fusion of bilateral cardiac crescent in the mesoderm. To have the fully developed heart from a linear tube, complex developmental process should happen, such as rightward looping, septation, valve formation, and so on. From now on, I will review such steps. At the third week after fertilization, initial linear heart tube develops from the cardiac crescent, and it is the first organ functioning in the embryo. This is a schematic drawing of formation of primitive heart tube. The bilateral part of the cardiac crescent fuse in the midline to form a primitive linear heart tube. The heart does not develop solely from cells of the primitive linear heart tube, which is called primary heart field. A second population of cardiac cells are present at the medial and ventral part of the primary heart field, which is called second heart field. The primary heart field gives rise mainly to the left ventricle in the formed heart, whereas the cells from the second heart field contributes to the atria, right ventricle, outflow tract, and systemic and pulmonary veins. Two extracardiac cellular populations also participate in the formation of the heart. The cardiac neural crest provides cells for the outflow tract, and proepicardial cells, which is located at the septum transversum, migrate to the surface of the heart and invade the myocardium to be fibroblast and smooth muscle cells for the coronary vasculature. The next step is conversion of the linear heart tube to be looped, segmented heart with multiple layers. The initial heart tube is lined by endocardial cells, which is basically the same as the endothelial cells of vessels. Later, outer myocardial layer develop. A thin endocardial tube is separated from myocardial layer by gelatinous connective tissue, which is called cardiac jelly. Uneven growth and expansion of each segment make a primitive atria and atrioventricular canal and ventricle outflow tract. As the volvus cordis and ventricular part grow faster than other lesions, the heart bends to the right to make a S-shaped heart. Heart looping is the first manifestation of right-left lateralization in the embryo. For cardiac chamber formation from the linear heart tube, segmentation model has been suggested as a classical model. Myocardium of a primitive linear heart tube is called primary myocardium, characterized by slow electric conduction and slow contraction. In the segment model, atria and ventricles composed of new working myocardium with fast electric conduction property align alternatively sinus venosus, AV canal, and outflow tract, which still has the characteristic of a primary myocardium. After several studies, ballooning model is more reasonable to explain formation of chambers. In this model, primary myocardium, colored in purple, originates from the linear heart tube, does not separate. It is persistent continuously, even in this stage, from inflow tract to outflow tract along the inner curvature. The atrial appendage and the apical part of the ventricles composed of working myocardium, shown in gray in these drawings, balloon out from the linear heart tube. The next 4 weeks, several developmental processes occur, such as septation, remodeling, and formation of a specialized tissue, including valve and conduction system. First is the atrial septation. Here is the primary septum, septum primum, colored in light blue, grows from the roof of the atrium to close the foramen primum. The foramen primum disappears, before the foramen primum disappears, perforation appears in the central part of the septum primum. The perforation fused to form another opening of the foramen secundum. After that, septum secundum, the secondary septum colored in green, forms at the right side of the septum primum as a fold of the atrial roof. It overlaps the foramen secundum, but it does not close completely to leave the foramen ovale open. The septum primum does not fuse directly to endocardial cushion part. There is another non-myocardial mesenchymal tissue growing from the dorsal mesocardium, which is called vestibular spine. Atrial septum is complete with the vestibular spine between primary septum and endocardial cushion. Originally non-myocardial tissue of the spina vestibuli is infiltrated by myocardial cells colored in black in these pictures. Here is the final arrangement of the atrial septum in the embryonic heart. Septum primum, septum secundum, and then vestibular spine, the low portion between septum primum and the endocardial cushion. This picture shows the atrial septum in the adult heart. You can recognize the inferior rim of the fossa ovalis is located at the same site of the vestibular spine. Before going to ventricular septation, let's talk about the development of pulmonary vein briefly. This is the bilateral atrium. The pulmonary vein opens to the left atrium, just left of the atrial septum in the mediastinal myocardium, developing later than the other part of the atrium. So, the pulmonary veins attain myocardial sleeves, which can be a substrate for atrial fibrillation in the adult heart later. The interventricular septum has three anatomical parts. the trabecular septum part, inlet septum, and outlet septum. The main portion of the ventricular septum, trabecular septum, is made of growing muscular ridge resulting from expansion of bilateral ventricles. There is interventricular foramen over the muscular septum. The interventricular foramen is finally closed by fusion of the endocardial cushion of the AV canal and endocardial ridge of the outflow tract. The fusion spot is where membranous septum is present in the adult heart. The inlet portion is formed by fusion and myocardialization of the AV canal endocardial cushion tissue, and the outlet septum is formed by fusion of the outflow tract endocardial ridge. So later, I will talk about the outflow tract septation in more detail. Outflow tract is composed of conus cordis to become ventricular outlet portion, and Truncus Arteriosus becoming intrapericardial arterial roots. This drawing shows outflow tract portion in embryonic heart connecting ventricles to the aortic sac. It has two endocardial ridges which are spirally arranged. When they fused each other, blood from right ventricle goes anterior and leftward to pulmonary trunk, and blood from the LV goes posteriorly and rightward to aorta. Ventricular arterial valves are formed at the middle of the outflow tract, separating outlet portion of the ventricles from the intrapericardial proximal segment of aorta and pulmonary trunks. These are the microscopic sections of the outflow tract in 6 weeks after fertilization and 8 weeks fertilization. Here, outflow tract septum is originally mesenchymal tissue. However, this septum is infiltrated by myocytes, which eventually become the muscular outflow tract septum. Even coronary opening has myocardial covering. Truncus Arteriosus is the situation of defective outflow tract septation, which is combination of a common trunk and juxta-arterial VSD. So this part originates from the outflow tract in the embryonic heart. TOF results from anterior superior displacement of the outflow tract septum portion of the proximal outlet part of the ventricle. Complete TGA has great vessels in parallel instead of normal crossing arrangement. It means outflow tract separation happens, but outflow tract ridges was not arranged spirally. Separation from the loop heart is not enough because atrium is connected to the LV, but not RV directly. Outflow tract is connected to the RV, but not to the LV directly. In order to have a direct connection of RA to RV, rightward expansion of the inner curvature is required. And for direct connection of the aorta to LV, leftward movement of outflow tract is required. DORV is an example of outflow tract remodeling defect. Both pulmonary trunk and aorta are still connected to the right ventricle. Finally, I'd like to review development of conduction tissue. In the linear heart, it is composed of a slow conducting myocardium, which has peristaltic contraction. The contraction wave always starts in the end of inflow tract, propagate to the outflow tract. The developing atrial chambers and ventricular chambers acquire fast conducting and contractile myocardium. The developing heart at this stage, even though there is no discrete conduction, it is possible to have an essential adult type EKG because of alternative arrangement of slow and fast conducting myocardium. As I mentioned before, the primary myocardium derived from the initial heart tube in purple is persistent in sinus venosus, AV canal, and the outflow tract, and inner curvature of the atria and ventricle in the developing heart. In the formed heart, SA node and AV node are derived from the remnant of primary myocardium, whereas the ventricular bundle branch are derived from the working myocardium. As I mentioned before, contraction in the developing heart always starts from the end of inflow tract, which is reflected by the location of a SA node at the sinoatrial junction in the formed heart. Atrioventricular node delays AV canal conduction for sequential contraction of atria and ventricles, which originate from the atrioventricular canal tissue made of slow conduction primary atrium. In contrast, the ventricular bundle branch derives from the fast conducting working myocardium for simultaneous ventricular contraction. This schematic drawings shows a working model for the developmental background of clinical arrhythmia in the child and adult. Selected clinical arrhythmias are frequently observed at the anatomic predilection sites, which may be related to the embryonic development of the cardiac conduction. For example, in the embryonic heart, myocardial continued AV canal may explain accessory pathway WPW syndrome. For example, myocardium surrounding pulmonary vein may explain atrial fibrillation and so on. That's for today's talk. Thank you very much for your attention.